Hey there, listeners. Welcome back to Let's Get Oral. Today, we are continuing our IDM Totality of Care series by sharing our conversation with Rachelle Beattie, a fellow microbial ecologist and the lead scientist at ProBiora Health. This chat with Rachelle is a nice continuation of our previous episode with David Lin from Bristol. Rachelle adds her insights on the role oral probiotics can play in maintaining and progressing oral health, and also how a better understanding of the oral microbiome is key for addressing oral systemic health holistically. Thanks again. So let's get to the show.
Let's get oral. Let's get oral.
Welcome to the show, Rachelle.
Thanks for having me. I'm excited to be here.
Yeah, I just wanted to pepper in that we were all at the IDM Society conglomeration with LMU, and so we're following up with Rachelle, who was a speaker at the event, and we were so busy at the event that we had to catch up with some of the speakers because it was a crazy fun time. Anyway, so first of all three of us are microbiologists by training. So let's just celebrate that fact, right?
Yes, very exciting.
So this is great because we cannot, but I guess just a reminder for us personally is to make it for the everyday person who maybe doesn't understand all of the technology or technological language that we use so cavalierly. But anyway, I wanted to hear about your journey as an ecologist and soil microbial, specialist, moving into the rural communities and then finally ending up in oral systemic health at an LMU oral systemic conference. How in the world did you make that journey?
Yeah, it doesn't sound like it makes very much sense whenever you say it out loud. I know whenever I gave my intro there, I was trying to make sure I could help people connect with how I got from A to Z because it's really about that far apart. But going in, even to my undergrad, I really didn't know what I wanted to be. I actually was going to school to become a dietitian. But they threw me into honors biology my first semester, and that pretty much solidified I was changing over to STEM. Loved the class, loved the lab, just got really excited by the things that I was learning there. Grew up in a really rural town in northwestern Missouri, right? So science there was not the most rigorous, to say the least. And so that first class really kind of changed things for me. But I think what really got me into ecology and specifically microbiology was actually my basic ecology course in undergrad. I remember when we got to the micro section, it was just one day out of the entire course. But the instructor was talking about how this was kind of an untapped field. I mean, we're talking back in 2012. Wow, that sounds forever ago now.
I know, doesn't it?
But just with how much the technology was changing and how much there was to explore, especially with sequencing technologies where we can go way beyond culturing with petri dishes and plates. But we can look at full-blown, full-scale microbial communities. And so that caught my interest. So I ended up getting a master's from the same institution I got my undergrad at, where I did a soil microbial ecology project. We were looking at the effect of lead contamination on soil microbial communities. And that really spurred my transition. I never thought I would go on to get a PhD, if I'm being fully honest. I'm a low-income first gen. I did not know what a PhD was. I thought a master's was higher because master sounds like you master the subject, right? And so this was all very new for me. But yeah, I ended up in Milwaukee, where I personally feel like I got really lucky to work with a great advisor, Krasi Rostova, where we investigated the influence of concentrated animal feeding farms. Milwaukee, Wisconsin, known for their dairies, right? Lots of animals, very small area. They produce a lot of poop. And typically, as a way to get rid of that waste, they spread it on crop fields as fertilizer. However, especially when you're in regions with a lot of snowpack, a lot of rainfall, or very fragile bedrock, you can end up with some of that manure waste in your groundwater. And if you're in a rural area, that can end up in your drinking water, especially in small communities. But also, a lot of, we have Great Lakes in that region as well. And so you can end up with runoff into those lakes too that people use for recreation. So that is that's what I was doing in my dissertation. I was looking at antibiotic resistance in the environment associated with these microbial communities. So there was kind of a clinical connection and component there. But as I was wrapping up, I was job hunting. I wasn't sure if I was going to do a postdoc. I didn't know if I'd continue in academia or not. But at that time, actually in late 2019, I found ProBiora Health. They had a very interesting job that I thought combined a lot of skills for my PhD. So not just the science, but also a lot of like the organizational skills that you learn and the management skills that you learn as a PhD. And I became really fascinated with their product, which was an oral care probiotic. Had never heard of one. At the time, I was probably like most folks, had heard of gut probiotics. But I really dug into their science, and what really caught my attention was that they were really using microbial ecology concepts, which was, how do the bacteria in the mouth interact with one another through competition or inhibition, making antibiotics or antimicrobials to help control levels of different bacteria in the mouth. And that's really the principle that they based their product off of. And I was hooked. And so
Absolutely.
I interviewed, worked with them on and off, full-time, part-time, full-time again now over the last few years while I explored a bunch of avenues, postdoc at UNC where I studied COVID and wastewater. I worked for the U.S. Geologic Survey for a while as well, which was really a highlight. But now I'm back with ProBiora Health, using microbial ecology in the oral microbiome. And that's how I ended up at IDM.
That's fantastic. Are you able to kind of make the leap back to rural communities in any meaningful way? Because we know that there's the socioeconomic issues in the rural communities related to oral health. Are you able to kind of bridge any of that?
That's really been a big push for our research. So with ProBiora Health, we're still actively pursuing lines of investigation of how our bacteria really work in the mouth, how they help improve oral health. And one of the communities that we are getting ready and gearing up to focus on are some of these lower income communities, especially like elementary schools. I lived in an area where there was one dentist for about 10,000, 20,000, maybe more than that people. I'm thinking of just the town, but the wider community as well. We all went to this one dental office. And I was lucky. My parents prioritized our teeth. I had 2 cleanings a year, but that was definitely not the case with a lot of folks that I went to school with. And so that's definitely a group of people that I'm passionate about and want to help take care of, but our company too.
Wow, that's great. I'm glad to hear that. I don't know how to bridge the gap for my own business, but I am definitely, investigating ways because I think we need to reach more people in a more meaningful way. They just don't have the tools or the knowledge, really. So that's fantastic. Do you look at the host side of the ecological system?
I have a little bit of experience there, but I'll be honest, no. I really truly have mostly focused on microbe to microbe interactions within an environment rather than how they work with the host. I can't, well, maybe not fully, right? Because we do a little bit in terms of like what that environment looks like, right? Which is controlled, at least for people in the oral cavity, a lot by what we do, right? What we eat, what we drink, if you smoke, things like that, the stress all influences that. So from the nutrient perspective, yes. But in terms of my brain always goes to like the bigger things that dentists and medical professionals are working on, right? Like inflammation and other factors. So not as much on that side.
Well, you mentioned inflammation, and I bring that up because
Mm-hmm.
I could talk about that all day. I
Yeah.
definitely worked on host microbiome, but my focus was how the immune system kind of tolerates or doesn't tolerate the microbial community, but also how it tailors. There's like a back and forth, obviously.
Mm-hmm.
So you have your natural antimicrobial peptides that are produced by the gingival tissues. You have your
Right.
inflammatory cytokines that are there even in health. So they are contributors always. And the microbial community, we know that some of these bacterial members do and can disassemble those inflammatory mediators as food, and that changes their whole metabolic profile to become more dysbiotic. And so some of the research that I've been following is to tailor the inflammatory response so that you end up with a more balanced community instead of always having to target the microbial community, because we know that broad spectrum deleterious microbiome challenges are just creating new dysbiosis. So I don't know if you're following any of that, but I would love to nerd out with you on some of those.
I love this. I feel like this is a whole new area that I want to read. This is my favorite part of microbial ecology. Like, you can get into just about any area, right? That's microbes are involved. They're interacting with their environment. They're interacting with their host. They're interacting with so many different things. So no, I don't know much about that, but just from your description, I definitely would like to know more.
Welcome home.
Are you? Yeah.
Yes, right.
Excellent. This is why we're all talking, right?
Right.
Are you perhaps going to be at AOSH?
I think I personally won't be, but I think our group will be.
Oh,
So
of course.
historically, we've had a long history and a good relationship with AOSH, which I think is important, right? Basic systemic health. So, yeah.
Well, I will be putting together a very lengthy talk on salivary. So maybe we could coordinate offline to kind of collab on what that might look like, because I'll be definitely talking about the host side, but I think, moving forward with discussing more of the, two-sided approach would be really a nice way to balance that very lengthy talk and make it palatable for everyone involved.
I love that. That sounds super exciting. Yeah.
Yeah. So you ended up at IDM. What were the biggest take-home messages for you from this conference? I know I have a very new, expansive appreciation for airway health, and was definitely noted. I think we all noticed, people sleeping inappropriately in the airplane afterwards on the way home.
Right.
And I feel like we should have coordinated a sleep study at the event, because I think now we're all going to go do that, obviously. But anyway, I wanted to know what your biggest take home was from all of the speakers.
I feel like I learned more than I have ever known, and I have a newfound appreciation for nitric oxide. Yeah. I absolutely loved the keynote talk. It's funny that you mentioned airway, because I just co-authored a short piece for the RDH journal about airway during pregnancy and how that relates to the oral microbiome. So that was really exciting.
Does it change during pregnancy?
It does change during pregnancy. Yeah. Definitely you tend to get an increase, particularly in gingival pathogens during pregnancy, but they think it's associated with the shift in breathing, right? So more pressure on the lungs, more pressure on the diaphragm, which can lead to pregnancy onset sleep apnea, right? And so you end up with a lot of those airway problems that they talked about, but that is very new for me. I really dug into the research for that, and I still have so much more to learn. But airway for sure. And then, the nitric oxide talk. It's funny. I listened to that one with my husband, who's a PhD, but he's a conservation biologist. And he was like, so where can we get, how can we add this into either our diet? We were waiting for Nathan Bryan to talk about the actual products, right? We're like, okay, we can hear the importance in this. So how do we get more of it? How do we help increase that production? So.
Yeah, absolutely. Really, I definitely have my bottle right next to me right now.
That's awesome.
I have the Oura ring for, everybody's information. Mm-hmm. I've been tracking all the changes. I would say that they're subtle, but they are present, which is interesting.
Yeah. Mm-hmm. Whit talked about that app that you could use for snoring. I don't know if you guys remember that. He talked about it during the opening, on the very first day. And he measured his and he personally changed a couple things over 5 days. He used some kind of tape. He used something to help open up his airway as well, and how that helps reduce his snoring over just a 5-day period. And I was like, oof, I think some people in my life could potentially use that app and maybe see how snoring's affecting their sleep.
Yeah, I think we were all in the same camp during and after IDM when it comes to how much attention we are giving to sleep and that sleep connection to oral health. And I mean, I don't know how true this is, but I feel like my Fitbit sleep scores over the last few days have actually been better, because I've been so mindful of all right, here's how I'm going to position my pillow. This is how, all the things. And I love it. So I'm on this journey with you all.
Is anybody mouth taping yet?
I'm
TBD.
not.
I do plan to give it a shot. I also just like do my due diligence because I like to play around with my facial hair dress up. So just wanting to make sure my mustache and beard's not being
I think
irritated.
there could be studies on this, but do you know of any studies that have looked at the microbial ecology in the mouth before and after mouth taping?
I do not. So actually, that was the first time I'd heard of mouth taping.
Okay.
And again, like I said, I think it's because I'm so new to the breathing area and how that affects it. But when he was
same.
talking about the mouth tape, I was like, people tape their mouths. That was very,
Mhm.
I was
Okay,
very surprised by that. So
I did go on like a sleuth of like what kind of brands are out there and which ones I could get that might breathe better than others. But there's a brand called Hostage, which is really hilarious. Yeah, exactly.
You're right.
Anyway,
Were there a
so
lot of brands then? So is that a pretty popular?
yeah.
Okay.
There's
Mhm.
lip-shaped one, which is great for me, but maybe not. If you identify as a man, that might not be your thing, or maybe that's what you do at night. I don't know. Anyway,
I feel like if
what
it
are
works, it
some?
works.
Yeah, exactly. I was going to start with just the surgical tape that I have at home that does breathe, just because it's on hand and right, that could
Right.
work, right? I don't know. Anyway, so back to the microbial ecology and ProBiora. What are some of the things that impact or improve the scores for the microbes in your assay there?
So our product is an oral probiotic, right? It contains 3 strains of Streptococcus, which I always want to emphasize. I know people hear the word Streptococcus, they think strep throat, right? But most
Yeah, I know.
Streptococcus, especially in your mouth,
Yeah.
they're healthy, they're commensal, they're meant to be there, and they're providing good benefits. But one of the big things that we see, well, a couple of things. There's really 2 mechanisms of action. 2 of the strains produce hydrogen peroxide, right? So that hydrogen peroxide helps inhibit the growth or outright kills anaerobic bacteria like P. gingivalis, right? Or Fusobacterium nucleatum that cause gum disease. So that's one big benefit. Hydrogen peroxide, people can get a little nervous about that, right? Because
Yes.
we used to use hydrogen peroxide rinses, which just kind of like antibiotics eliminate almost everything in your mouth.
Right.
The level they produce is pretty low. I think if I remember, it's somewhere, it's less than a fifth of what you would get in a rinse. So it's enough for that microbial inhibition and to gently whiten your teeth, which is like an added social benefit, right? Because bright white smiles can help improve confidence.
Mm-hmm.
But not so much that you're going to eliminate or destroy the vast majority of your microbiome, causing dysbiosis. The other strain that's in there, Streptococcus rattus, is it's my favorite one. It's a natural mutant of Streptococcus mutans, right? We all know about mutans. It causes cavities.
Yes.
But the special thing about our rattus mutant, and it's a natural one, it was just like developing antibiotic resistance. It was plated out, right? Discovered to be missing the gene to produce lactic acid. So it competes for space with strep mutans because they use the same nutrients. They live on the tooth. They inhabit the same ecological niche, but one produces acid, mutans causing cavities, one doesn't. And so, overpopulating with that rattus strain can help lower the numbers of mutans and help lower the numbers of the strains that are producing acid on the teeth and hopefully help reduce cavities.
Is this the same strain that's been explored as a replacement therapy for those who suffer from caries?
Yeah, it is that strain. And we have quite a few good studies, particularly in children, right, who are generally more prone to cavities, especially in those early teeth, showing that it significantly reduces the levels over between 30, 60, 90 days. But that effect lasts, even if people stop taking the product, that effect can last. I think the longest that we've looked was over 3 years. So it's pretty cool. Yeah.
That's fantastic. I love the longevity trials. I think that is the most important and often missing piece of all of the clinical work that we've done in the probiotic space. So I really appreciate the effort there. I know it's not an easy task to follow up after so much time. Getting people back in for an assessment can be real challenging. So how do you do that? Do you just have them send samples from home, or do you make them come in and
I'm going to be honest, a lot of this work was done pre-me.
Okay,
That
all right.
work on the follow-up and the initial study was done by Mark Cannon, who was affiliated with the University of Chicago. I think I've got
Got
that
it.
right. Northwestern. He was affiliated with both, actually. He did that study, and we've had a couple of other dentists over the years, but that's who we typically rely on, right? Because ProBiora is a pretty small company. We still consider ourselves a startup, and
Of course.
space-wise, things like that, and lab-wise, I just got my own lab here. We're getting it set up right now, so that's really exciting to come back into the lab and to get more basic studies as well as clinical studies going in collaboration with some dentists. But yes, I know from speaking with him, especially about that study in particular with the follow-up, he was very excited with the return because they had 60 participants. And if I remember right, it was in the 50s. I want to say it was 54. They got the 3-year
Wow.
follow-up. And
That's
that was huge. Yeah.
That's
huge.
like a miracle, honestly.
So
Wow.
I know. Yeah.
Yeah. I mean, congratulations. I hope you can get that again, because those are the most important pieces that we're all trying to follow up with. So that's fantastic.
Yeah.
Jonathan, do you have any follow-up questions?
I love that y'all done the work in science on particularly a lot of these strep strains. And I say that because Camille does this, but Rachelle, you probably don't. During my PhD work, I actually investigated some of these strains in a biofilm model I developed to understand interkingdom interactions between commensals and either caries or periopathogens in relation to their relationship with Candida and other fungi. And I think that's been a particularly piece where, like, while Candida is opportunistic, therefore, like, typically not an issue for many of us, but for those with autoimmune or aging populations, people deal with dry mouth. All it takes is one variable to make them a long-term problematic issue. And what was always fascinating to me was how complex these relationships were when it comes to cross-kingdom interactions, because a lot of these streps you pointed to, in my model at least, a lot of those relationships seem to be actually synergistic, which, if you take a step back, it's kind of scary. Like, if you grow these two bugs together, they both grow at like faster and greater, and they utilize metabolites and nutrients more quickly. But getting back to when we were talking about nitric oxide, and I think about the nitrate and nitrite reducers of the oral microbiome, they played a such key role in modulating and fine-tuning the growth and a relationship between Candida and a lot of the strep species that seem to have a synergistic relationship in a two-person model. So my question to you is, have y'all explored more complex communities and explorations with your probiotics and other platforms?
This is such a great question. Love this question. We are in the middle of a study right now where we're actually investigating, because of that kind of research, where we found that some commensal streptococcus actually can increase their growth and candida growth, right? Whenever they're grown together. We're in the middle of an investigation on this right now. So results pending. I'm not sure exactly what that will look like. But I think one of the big things that I've learned, particularly when it comes to these complex communities, is that relationships in the lab don't always, and I know you guys know this, they don't always mimic what's actually happening in the mouth, right? Because there are so many other species. And so I like that you mentioned that you guys investigated it as like a 2 species, but then also that nitrate and nitrite reducers can influence that as well. There's a lot of pressure, and we do a lot of reporting on like single species, dual species studies, as if like this is exactly what's going to happen in the mouth as well, when in reality, if we're in the science, we know that it's not. But whenever it's being like pushed out to the public, they're hearing the result of just that one study without, and rightfully and understandably, without knowing like the full context of what that means. But so yes, we are in the middle of that investigation, because we had a couple of people, Mark Cannon being one of them, come to us and be like, "Whew, have you seen this research? It looks like Streptococcus aureus increases levels of Candida, and whew, let's panic." And that's good. I mean, that's what we want to see, because obviously, I mean, the company's goal, of course, is to get probiotic in people's mouths as a health benefit. I mean, and of course, companies as a whole, their overarching goal is to make money. They're a business. That's the model. But as the scientist on the team, it's really to make sure that we're putting the best thing possible forward to help the most people, and that we're putting something out there that's actually going to help improve oral health, for the betterment of overall health. And so I think it's important that we continue these studies and don't panic whenever we see something that might not look that potentially can look bad, but that you have to explore it further. So I love that. I would love to know what all you guys have found.
Yeah, I want to interject one thing there. Okay, so Jonathan, you're talking about bridging communities. So we think about F nucleatum to be, a potential periopathogen, but it's a primary player in making sure these nitrate reducers are present.
Yes.
And we also know that some nuke strains are worse than others or better than others. And so there's a lot of room for discussion and room to allow for some of these species to be naturally available in small numbers. And I think the same might be true for Porphyromonas gingivalis, because it does degrade the inflammatory mediators. So you could see how in very small numbers, it might be just fine and it might be providing that benefit. But again, it is a numbers game, and dysbiosis and really understanding that all of the nuance there is tricky, and we all have to kind of get creative. Also, I wanted to point out that Candida, however you want to say it, there is evidence that with the innate immune system, it is teaching the innate immune system. It is part of how we inform our innate immunity how to behave correctly. And if you're never exposed to fungal communities, and Candida is actually a commensal, but again, in smaller numbers, and it plays a huge role in priming the immune response to regulate appropriately. So I think, all of this is good. And to get mired into like one is bad and evil and we should eliminate it is a mistake. And we need to kind of allow for a variety of activities that are balanced with the host. And that's why I'm always, hounding on the host side, because we've just been anti-bac for so long in the mouth. And I just love that we're creating a community that is appreciating the nuance.
Yeah, I just learned something about P. gingivalis. So that was super exciting. And one of the things I talked about at IDM is that, it is there in low numbers, even in healthy mouths. And I think that was a question for a long time. It's like, oof, if it's only causing disease, what is it doing in these healthy mouths? So what a good fact that I just learned.
Exactly. I've been saying this for a long time, and everybody cringes when I come into the room. They're like, oh, it's that. Oh God, you're going to tell us PG is good. Great.
Right.
But it will change its metabolic behavior depending on how much inflammatory cytokine load is there. So, it's very nuanced discussion. And I'm not saying that it can't be the worst guy on the planet at times, But again, we do have to appreciate some of the benefits and why we've evolved with these commensals for such a long time. They're certainly not all bad to whatever degree they're supposed to be there.
Yeah.
Right. And I also love that not only do y'all think about we the people, but y'all also have formulations that target our fur friends, or the cats and dogs.
Yeah.
And so I'm curious to know, because one of our previous guests, Holly Gant, she actually is she's a microbial ecologist, so part of our community. And I'd be curious to know, like, A, do you have any pets?
Yeah. Oh, yeah, I do. I have 2 dogs. One is laying next to me right now. I'm very proud of them. They're being quiet at the moment. Normally I get on a call and they want to play fetch immediately. I have a red heeler mix, and then I have a corgi mix as well. Yeah. So I've got 2 dogs.
Nice. And are they test subjects for your probiotics?
They are. So it's funny. When I was in grad school, I only had the corgi mix. She's almost 8 now. And oh my goodness, she had the worst breath. I know, like, my healer mix, nothing. He's perfectly fine. Like, he's always had pretty good breath, but she just, I don't know what it was. I joined the company, started using the product, right? And immediately noticed a change in her breath. And I'll be honest, I was kind of skeptical about the fact that we use the same blend. So the blend is the same, right? So the same 3 strains that are isolated from human mouths. And a lot of people, if they're kind of like savvy about the microbial community of mouths, realize that canine and feline mouths are very different. Their microbial communities are different. The species that are there are different. So we get asked a lot. We're like, okay, like, why are you putting these human strains into a pet mouth? Are you sure that it works? Things like that. Those are all really good questions. But it turns out that they can survive. So in your pet mouth, they can, just like in a human mouth, right? So they're able to attach and survive. Streptococcus species are actually pretty common in dog mouths. and that they're performing those same functions, but instead of like preventing things like cavities, right, they're really helping with those bacteria, helping reduce levels of the bacteria that either cause things like gum disease, which can lead to the really stinky yellow breath, yellow teeth, bad breath, or bacteria. I'm like staring at my dog now as I talk about it. He's chilling right here next to me. Or the ones that cause things like halitosis, even in your dog. But yeah, they've been great test subjects. They love the product, so I got pretty lucky there. And her breath is significantly better, so that's really nice too.
That's a win-win.
Yeah.
That's fantastic. Yeah,
she's 8 and she's never had a cleaning, so I'd say, that's pretty good.
Yay, that's money back in your pocket for more treats and pet adventures.
Right.
Yeah, I wasn't put on with regards to pet oral and gut microbiome until the last few years, and just being more attentive to their holistic health and using diagnostics and whatnot to cater to it accordingly. But the parallels between us and them when it comes to that science is undeniable as I further dive into it. I am getting back to oral systemic health and IDM. I would love to hear your opinion about how you envision the role of the oral microbiome pushing the field forward. Do you think it's going to be more diagnostic driven or therapy driven or a combination of those things? How do you envision it?
I know you guys had Laura Hooper on recently, and she is I've known her for a couple of years now, and actually she's like my presentation idol. Man, she talks and I just want to listen.
Yeah, she's wonderful.
She's one of those, and and she's very science backed. So she's not just a good speaker who can convince you of anything, right? Like she's a hygienist who's so involved in the science. It's beautiful. But she is really the one, she's where I learned that concept. I think she says test, treat, repopulate, right? She's kind of got this like trio of things. And I think that's really where the microbial community comes into play here in oral systemic health. Because I really like the model of you need to know what's there first, right? Like we can't treat everybody like they're the same. Individual mouth, the way that it carries the disease or responds to a treatment is going to be very different between individuals. And so there's not a one-size-fit-all solution, but the starting solution I really do think is salivary diagnostics. So let's take a look at what does your oral microbiome look like. And then in addition, salivary diagnostics is expanding into like inflammation markers and other things as well. But if we're just talking microbiome, let's take a look at who's there. And then do we need to eradicate? So, obviously if we're testing, most likely there is some disease state. If we need to eradicate, what can we use to eliminate the problem pathogens rather than the whole microbiome? So we're trying to boost those commensals, followed by repopulate, right? And I think that's where probiotics or potentially prebiotics. I think I was a little bit harsh on them in my presentation on IDM because I think we've really studied prebiotics for the gut. How do we help foster a healthy gut microbiome? There's a beautiful paper that just came out actually like a month ago that I was just reading yesterday talking about prebiotics in the oral cavity. So that's exciting. I think a lot of them had to do with nitric oxide. So that's exciting.
Yep. I know exactly what you're talking about too.
Yeah. And so I think, that makes me happy. I like to see that a little bit more. But yeah, how we can both promote health in our native microbiome, as Cami would have said, but then also if you need a replenishment, right, of some of those beneficial bacteria, what can we get in there? And I also don't think that's one size fit all, right? We're just one product with 3 strains, that do some variation of work. If you have a lot of P. gingivalis or too much FN, right, our product is probably a pretty good fit, but there might be situations where it's not. So
I'm glad that you bring up those nuances in terms of like in the name of being able to identify, monitor, eradicate if necessary, depending on the state of health versus disease and the association with the potential pathogen, and then repeat, right? in terms of the monitoring surveillance. It's especially since the mouth has such a high turnover due to our hygiene practices and just this nature of the mouth in and of itself. It's very unique compared to other parts of our body.
Yeah. I was really fascinated. I just recently learned about the shearing force of your saliva and like just how strong that shearing force is, which means like attachment of bacteria on your teeth. That's why that biofilm and that plaque is so sticky, because they literally can't if they're not strong, just swishing the saliva around your mouth, the movement of your tongue can just knock them right off. I was like, oh my goodness, they have to have such strong attachment, to be able to, because you need a nice healthy biofilm on your teeth.
Mm-hmm.
So yeah, it's really interesting.
Yeah. The one of the aspects of my thesis project that I first focused on was cell adhesion, and then how that looked on different materials, whether it was like enamel, titanium, and other dental brackets, dentures. And that, I think that's what scared me. The truth, when I was like, it's only been 3 hours and I see how much growth, and then to try to clean those substances, like brushing wasn't doing it. I was like, do I need to get a sonicator in here and disrupt some of these biofilms?
What do they attach to the best? So is it to enamel, or is it to some of those like dental appliances or implants?
So this is where it depends, right, on the consortium. Okay. And right, like, for transparency for people who are listening, this was all done in vitro, right, using a biofilm model, but some of them getting as complex to 15 plus different organisms.
Yeah.
And I would say what I saw more consistently was if you had Candida present and it was capable of morphing into its hyphal form, meaning it goes from a singular yeast cell to more of filamentous format, it was almost always hella sticky to denture-based materials and also to enamel and enamel-like materials in my assay. Wow. And what was wild about it was how quick that growth would be, or overgrowth would be, especially when it was paired with various strains of Streptococcus mutans, Strep gordonii, and a few Strep aurealis strains that I investigated. Yeah. But on the flip side, when you would introduce various Veillonelli into that consortium, you would not see hyphal growth. You would see very little to no Candida growth. Huh. And I would see a lot less. You'd still have stickiness, but it would be not near as sticky. And that is really cool. And the key thing I didn't probably mention to this is probably a lot of these biofilms were done strictly under hypoxic or anaerobic conditions. Okay. Because I was really wanting to understand what's happening deep into those pockets, or those tight crevices and deeper parts of the biofilm. And for your strict anaerobes, right, it makes sense for them to be able to thrive in there. But once I started pulling them out of the hypoxic environment, it was amazing how well Candida could potentially provide protection or local hypoxia to those parts of the biofilms, because they are facultative and able to utilize oxygen and then share the nutrients that they're producing. Because one of them is like, oh, they're producing lactate. And guess who uses lactate as a carbon source? Veillonella. Yeah. So it was really fascinating seeing how those communities, as it became more complex in my model, would be associated with various levels of adhesion on these materials. And then again, how the environment, whether it's hypoxic or not, affected that accordingly.
Did you have like the more diverse, the less sticky overall? Did you see that? Right? Because I think that's something they kind of, did you? Okay. Well, that's good. Yeah.
Generally, right?
Generally.
Yeah.
Okay. Yeah.
It just became, no pun intended, really sticky though. Once it was like, if it was Candida strep mutans, it was like, that was the scariest thing to see in a lab. And especially when you're looking at, like, what's the expression level of some of these like key genes that are tied with adhesion or tied with pathogenesis. And I was like, oh my goodness, they're just like flashing red on my qPCR and whatnot. I was just like, my goodness, is this what's happening in our mouth? Like, TBD, right? But it was hard to not be fascinated though with the complexity and nuances of those relationships. And so that's what really put me on to thinking about the importance of monitoring and surveillance for children. And we talk about early childhood caries, because the research is clear that it's not uncommon for those lesions to have Candida present or being co-isolated with aggressive strains of Streptococcus mutans. And so, like, fortunately for kids, at least, there's still time to be able to correct that accordingly. But yeah, again, how do you do so in a way that is balanced and not just fully eliminating, some of our good commensals or the keystone microbes that are present.
Yeah, that is the big question, right? Yeah.
Do you, between your research and just like your life story, I think how it ties into this is fascinating. Do you have any observations about how our human behaviors over the last few years may have contributed to some of these developments where we might need to have oral probiotic supplementation becoming more commonplace for certain populations?
I think that is such a good question. I think, I mean, obviously we had a huge shift during COVID, right? When everything shut down. So couldn't get into the dentist, or you were scared to go to the dentist. Mostly, I think for the most part, people just couldn't get in, right? That's definitely one of those areas. If you can stay out of people's mouths, you're less likely to get sick. So we just stayed out of people's mouths. And I think that that's a situation where, I think my answer is kind of nuanced. I have a big passion for so I think that is a good example of how the last couple of years have changed like our perspective, right? People are just now going back to get dental care. They might have had a long period in between. And I think anything that you can do in that time to help support your oral health is beneficial. But keep in mind, a probiotic might not always be the answer, right? And so it really takes going in and getting tested to see what's going on before you make that move. Probiotics can obviously help support health, but they can't eradicate a disease. They're not a drug. They're not going to fully eliminate a problem if it's already really set in and is quite progressed. So I think that's one thing. They can be probiotics can be a really nice adjunctive, right? If you don't have an alternative option, brush, floss, probiotic, I think that's helpful if that's all you've got. But I will say for me in particular, I think obviously my passion and one of my focuses is in areas where, there's a lot of socioeconomic pressure, whether that be low income, rural in particular, where you just don't have a lot of access. And I think that probiotics can be a good alternative option. But I will say that right now, I still think that there is a money barrier. They're not covered. They're not covered by insurance. It's not something that I'm unaware of. I think sometimes, for my team, it might be a little bit difficult for them to listen to me because, I'm super passionate about the regulations, how we regulate probiotics. Are they actually safe? Are they actually effective?
Right.
Do you know what's being put into your body? Because, my family, they've got me, but how many people don't, right? And they're not somebody who could go and pick up a probiotic bottle off the shelf, read the strains or the species or whatever's on the back of the bottle, because sometimes it's not even at the strain level, look at the CFUs and be able to tell themselves if that's going to be effective for whatever they picked it up for. Yeah. And I think that just really complicates the use. And so for me, I think that's the big thing. I think more education is needed. Probiotic has also become a buzzword.
Yes.
So I hear a lot of professionals that are like, probiotic, isn't that just kind of like
another marketing?
Science?
Yep.
Yes, exactly. And what's crazy to me is there are people that are studying bacterial strains, but calling them something completely different that's more like friendly in terms of science is biocontrol. We've been using biocontrol in the environment for ages. And in reality, if it's providing some kind of a benefit, is that not also what we would call a probiotic? They're doing it. Marquette got a grant to study using different bacterial strains to help improve wounds in the field, so wounds for soldiers, right? But that's the same thing. It's a bacteria being applied to provide a health benefit. So again, isn't that a probiotic? But what we hear, like what you said, is it's just a marketing ploy, And I think that's kind of a very round answer. I think that's really where the struggle lies. Is I think there's a monetary barrier still, right? It's typically marketed towards middle upper class folks who can afford to add it as something into their lifestyle. When in reality, a lot of people probably truly could benefit from taking one if it was accessible.
Right. Yeah. The money barrier is definitely a persistent challenge. That's where I think education is going to be key for helping to, push the needle forward for breaking through some of those barriers.
Yes.
And then getting back to that, my question now, some of the other behaviors that whether you're using probiotics or not, that I think has paired to the state of the state today, especially since the pandemic. I'm not a foodie. I am an eater, but it is clear to me as someone who loves to eat that I feel like we societally are terrible about chewing our food, right?
Oh, yeah.
Mastication and the role that plays, and then how that can contribute to turnover, producing saliva, the sheer force of saliva. like just the benefits for mouth health outside of the microbiome, but also the interplay it has with promoting a balanced oral microbiome. So not ironically, but like to that point, that is where I am a huge advocate for probiotics as necessary, or prebiotics, or other types of formats, whether we're talking about gums, tablets, things that encourage people to chew, right?
Yeah.
And
Yeah.
do some mastication, because a little goes a long way.
Yeah. You know, I really think like diet and lifestyle do play a huge role in this too. And I think, you know, you look at the American diet, people always talk about it. They always rag on it. It's so processed, right? So much processed food, so much sugar. And I think after a while, people get really tired of hearing that, right? They eat what's accessible.
Yeah.
You eat what's accessible, what's in your area, and what you can afford. But it's also not necessarily wrong that if we could get more like whole foods, more fiber, things like that into your diet, you are going to help improve the native state of your microbiome, right? Which, you know, might be another. I think our American diet is a good reason to incorporate a probiotic, right? We're definitely missing some things. So that's another lifestyle factor that I think definitely plays a role there, and when it would benefit.
I'm curious to see over time, like as these technologies, as we just get more clinical studies, long form studies, and a better understanding of these associations, how that impacts the various formats, right? Because, like you said, on the market, like we're seeing beverages, you're seeing the term probiotic and prebiotic added to everything, but how many people who are buying and using those substances and products know if it's actually doing anything to improve their oral health or their gut health at a molecular level, right? Like that's not to say that like, hey, like if my bowel movements are getting better after taking this, like I'm not saying you can't have some sort of observation to see good benefits, but it's, as you know, like one solution does not fit all of us because there's diversity and variability in that. So that's where I think the parallel with the complexity of the oral microbiome, how it pairs to its connection to the gut and the rest of the body, and again, our own life scenarios and situations is key for having that monitoring aspect, that diagnostic aspect, and being able to take that long term to better understand what fits each person well or each community well.
Right.
Another aspect of the oral microbiome that I appreciate there are some researchers out there looking into this, but I think there's still a lot more work to be done, is the relationship between taste and flavor perception and how oral microbial communities may play a role in that for each of us. Because there's a spectrum of tasters, as you know, but I think most people don't realize the role like bitter taste receptors play in immunity. And lo and behold, our bugs like PG and whatnot can interact with those receptors in a way that toggle molecular mechanisms in real time. So I'm curious to see if there's more work to be, or more research to be understood, if you're a super taster or a normal taster, or someone who's doesn't have the greatest sensitivity to, say, bitterness, if there's different associations or relationships between how their oral microbiome may look in those specific sites, and then further down, has that impact their health status.
I love this. This is not something that I have not talked to anyone about this before. I have not thought of it before, but also, so if that plays a role, it's going to affect what you eat, right? Like, which is what you just said. It's going to affect what you eat, which is going to affect. Ooh, fascinating. Can't wait to see where this goes.
I mean, not to make this a philosophical episode, but it's just like, do we have free will in our taste, our eating habits?
Right. Or is it controlled by our oral microbiome, right?
Exactly. So on that note, what's your favorite type of foods?
I can tell you I don't like cilantro, so, you know, but hopefully that's just a gene thing, but maybe that has to do with my microbiome. I don't know. I'm not, you know, that's funny though, because I'm not a big sweets person at all. And I don't struggle. I feel very lucky. I'm going to be honest. My parents took good care of my teeth, right? They took us in for the cleanings. We got the checkups, and I have discovered I actually had a pretty progressive dentist who, you know, for kids, when they talk about plaque on the teeth and getting rid of all the plaque and all the things you can do, make sure just you get rid of everything. My dentist was very much like, do you know what plaque is? it has to do with bacteria producing acid, doing these things, causing these interactions. And I mean, I remember hearing about that when I was like 8.
That's fantastic.
Didn't realize until much later that so many dentists, well, one, they're busy, they're packed. They're coming in, they're doing what they have to do. They're getting you out of there.
Mm-hmm.
But they're really just not talking about what's actually going on. A lot of people don't realize plaque, is a bacterial biofilm, and all the different things that are happening in there, and the importance of keeping at least a healthy biofilm on your teeth and stuff. So, but I wonder, I don't know. I don't like sugary foods. Is that why my teeth are healthy?
I think that probably plays a big role into it. As someone with a sweet tooth, and I have the damage to show what that has done to my enamel over the years. And it's not to say my dentist, I had my dental checkups and whatnot. They're like, dude, stop eating the candy. I'm just like, it's not always candy. I was just like, I grew up playing sports, so I was sucking on oranges in between practice and drinking Gatorades.
Those little cuties.
Exactly. And I just happen to like sweets too.
Oh, yes. So what role does the microbiome play there? That is a good question.
Mm-hmm.
I love that. Yeah.
Yeah. I think another missed opportunity, and I mean, there's technologies and some practices that do this now, but like at chair side, especially for people going for cleaning, it's like we have very low cost tools and resources to be able to show someone's biofilm in real time.
Yeah.
Whether it's you do a scraping, putting on a slide and looking under the microscope, doing some staining. I feel like there's some ways to be like, hey, this is what plaque biofilm looks like, right? And that, they're like, but I brushed my teeth this morning. And you're like, yeah, exactly.
Right.
And look at what has happened since then. I think those little efforts can go a long way for sticking with people and be like, oh, maybe I should be more intentional about brushing after, 30 minutes after a meal, and especially brushing at night, because there are some people who just brush once a day, right?
And it's in the morning. Yeah.
Right.
Yep.
So I hope that if there's any practitioners who are listening and they're curious, feel free to reach out. I can definitely share some ideas of how you can incorporate such tools in your practice or facility to scare the children. Okay. On a less serious note, but as a microbial ecologist, I think it would be silly if I didn't ask, do you have a favorite microbe? And if so, why?
I had a feeling I was going to get asked this question, but you were the first person to ever ask me that. I thought about this years ago. I was like, I need to go into a classroom if I ever get asked that, and students don't care what your favorite microbe is. I struggle with this because I love so many. For a while, I was really fascinated with purple sulfur bacteria, and I swear it was really just because of the color, but also the reactions that they do fascinate me. But okay, you know what? I'm going to back this up. If we're in the oral microbiome and I'm thinking of a favorite in there right now, I do have a personal favorite, and it is one of ours, and I think it's just because I've done so much research on it and I've learned so much about it, but I love our Streptococcus rattus JH145 strain. Mostly because I find like competition between microbes really fascinating. But the fact that, we have a stable mutant that doesn't produce lactic acid that can outcompete, its parent strain strep mutans for spots on the tooth, right? And it's as simple as that. It's such a simple interaction. And for me, it's really important. If you're going to use something like a probiotic, you need to know how it works, not just, hey, we did something and it looks like the levels went down, so maybe it reduces. Do you know how? Like, it's important for me. You need to know how. And so I think that's probably my favorite.
I get it.
Yeah.
That's cool.
Do you have a favorite?
Oh, of course. I would say, it's probably a boring one for some, but one of mine's Saccharomyces cerevisiae. Just
that's not boring.
Right?
That's great.
Like if you enjoy beer, bread, just like some of these goody things, like that was a key one. My current favorite though right now is Aspergillus oryzae, or the one the fungi responsible for making sake. Another name for it is koji. I think it's it's been commercially utilized in various industries, so it's definitely been hella studied and leverage in many ways, but I love using it when it comes to doing like kitchen-based fermentations
Yeah.
for either meats or like doing a rice koji. And I'm also like currently doing some fun experiments in my kitchen on seeing if you can use it for upcycling waste.
Yeah.
So I'm a huge fan of that microbe and all the various like enzymes and metabolites it's able to produce and release, and in a
Yeah.
manner that is hella easy, right? Like it's like I just throw it in a tub and I come back 2 days and I'm like, voila, it smells fantastic. I love the smell of my kitchen right now, and this food's gonna have some funky flavors to it that the average person would be like, oh, how'd you do that? And I'm just like, it's just mold.
See, I love this. Yours are useful. And I'm sitting over here thinking, maybe my actual favorite one is that bacterium. Is it Actinomycetes in the soil that gives you that smell after rain? See, yours are useful. And I'm just over here just like, they look cool. They smell cool. That's why I like
they're all important though.
Right. They all give us some kind of joy, right?
Yes. Well, Rachelle, I really appreciate you taking time out of your day to be able to connect and sit down with Camille and I here at Let's Get Oral. We definitely would love to stay tuned in and follow up with you for future episodes if you're open to that, and definitely connecting with you offline to chat a bit more on this microbe stuff. But if you have any advice or any wisdom you'd like to share for our listeners, could you share?
Well, I think since this all kind of started, one, thank you for having me, Jonathan, and thank Camille for me too. I really appreciate that. This has been really fun. And since we started with, kind of the oral systemic connection, I think just like if you had one take home from the whole IDM symposium and everybody that you guys talked to, or even this interview, is just really that, your mouth is really the gateway to the body, literally, metaphorically, everything kind of starts there. And so taking care of your mouth is really important. So do what you can. Make sure you take care of your teeth, take care of your oral cavity and your oral microbiome. And that should really help support your overall health.
That's fantastic. Thank you so much, Rachelle.
Thank you.
Let's get